We are going to have an important presentation in this plenary session. The presentation will include two eminent professors from the National Research Center. They are going to speak on the use of nanoball as a potential treatment technique for patients with cancer. I hope that we are going to have a fruitful discussion after the two presentations. Now, Professor Shifo.
It is also a great pleasure to have first speaker, Professor Grant Professional, laser group system, Excellency Spectroscopy Department in the National Research Council. And the second speaker after him will be Professor Ahmed Abdul Omnishi. Professor Ahmed, please, floor.
What's nanomaterials? Nanomaterials are materials having at least one dimension in the nanoscale. But typically we call nanomaterial as the material which have only at least only one dimension in the range 100 to 1 nanometers. Nanotechnology is the manipulation of matter at atomic and molecular scale to create materials with new and advanced properties. This definition reflects the fact that quantum mechanical effects are important at this quantum real scale. This leads to accumulation of the efforts from physics, chemistry, biology, medicine, and engineering, and other fields to help to advance quickly the world of medicine. Gold nanotechnology is important in cancer fighting. Cancer cells, size, morphology, functional groups on the nanoball surface and cell types determine the subcellular distribution of nanoball in it. The different actions can be placed by the nanoparticles by photothermal destruction or mechanical or others. There are many different shapes of nanoball, including nanoparticles, nanorods, holospheres, stars, and others. Nanoparticles are emerging as promising agent for cancer therapy and are leading to drug delivery, photothermal agents, and contrast agent, and radial sensitivity. Nanoparticles can work as a nanodevice since it can be introduced into the cell itself due to the very small dimension of the nanoparticles. The action can be photothermal, localized surface plasmon resonance, or colloids. Both colloids also are important in this field. Nanoparticles can work in diagnostic and therapeutic applications. You are interested only in therapeutic applications. Due to the small size of the nanoparticles, we will deliver some energy to help the nanoparticle to kill the cells. So the response of the cell and nanoparticle must be suitable with fitted with the laser power. So the laser wavelengths must be matched with the nanoparticle absorption, so that the laser power delivered by the laser beam can be absorbed by the nanoparticles, then it kills the cells. The cellular interaction, once the nanoparticle has safely transported to the targeted organ, it binds to the tumor cell itself. So it is selective treatment. And nanoparticles can enter the cell and transport the energy to kill the cell when we expose laser to the cell. In fact, we have the biological window which can permit or dismiss the laser to enter to the nanoparticles. So the biological window here appears in the range of wavelength from 650 to before 1000 nanometers. So this is the wavelength range which we can use the laser beam to affect or to heat up the nanoparticles. This is the depigration of nanoparticles. Nanoparticles of metals like gold are inactive, so we have to make it active, biologically active. So it is pegylated by polyethylene glycol coating. This is the process of preparing the nanorods of gold. We start with salts dissolved from gold, then to start seeding, and to stop the seeding at some instance, then to start coating the nanoparticle with some material to make it biologically active, then to diagnose it. And here I give you some information about our work in the National Research Center related to nanoparticles treatment with laser. Here we used nanoparticles of gold to treat lab animals, and we expose the animals to laser, to first to cancer. We got the animals cancer, then we started treating. Here we were, this work started 7 years ago. This is the crystallization of nanogold. It is carried out using transmission electron microscopy and UV visible spectrometry. Here we can see the absorption of the nanoparticles suitable to the biological window, and the nanoparticle size is small and homogeneous. Here we give the histology of the cells. So here nanogold particles coupled with photothermal therapy showed massive tissue destruction, appeared as non-cellular drips. Here the scale is very small, 20 micrometer. The activity of nanogold coupled with laser and with photos thermal interaction. Is here, in this case, we checked the two types of injection, intratumoral and venous injection, intravenous. So we found that both are similar in exposing the nanogold sample, nanogold material to the cancer cells. Here we repeated this for experimental animals 10 times, and the mean here is for 10 replicates. And here the distribution or the presence of nanogold particles for in tumor itself, and liver, kidney, and spleen. So here we find with time the content of nanogold is decreased. This is the this was the kinetic pharmacokinetic study. From that work, we can conclude that gold nanoparticles demonstrated systemically are equally distributed and similar to that intratumoral administration. The accumulation of nanogold in vital organs such as liver and spleen appears to be non-toxic. However, we have to continue. This one of these studies started long time ago, and we continue to explore for different experiments, and the toxicity study in short term and in long term also on experimental animals was carried out, and the results showed no significant difference between the treated animals and normal animals after long time, after 1 year. The presence of nanogold in the animal is normally in the normal range, in the fluctuation of individual variation. And another example on big animals, and Professor Ahmed Abdul will speak about it in details, and it is published a few months ago, 1 month ago. And internationally, the gold nanoparticles appears to give alone with no treatment similar to that with gold only. So not nanogold particles itself is affecting the animal or the tissue, but the irradiation plus nanoparticles of gold gives the effect. This is well known from this figure. And finally, I'd like to speak about the advantages of nanotechnology. Here, nanogold are excellent tool for cancer cell imaging and basic research. However, they have yet to reach their full potential in clinic. They protect the drug from being degraded in the body before they reach their target. They enhance absorption of drug into tumors and into cancerous cells. They allow for better control over the timing and distribution of drug to the tissue. They prevent the drug from interacting with normal cells, thus limiting its side effects. And the drawbacks, the first one is, if we are dealing with nanomaterial in the solid form, it is extremely hazardous, like exposing the lung to asbestos. But we use the preparation in the liquid form, so we avoided this drawback. Gold nanoparticles require high purity materials as well as highly specialized manpower. They require also expensive investigations in dimensions, shape, homogeneity, sterility, reproducibility, and the clearance of the drug from the body is still having investigations, have to be investigated more and more. Presently, nanotechnology is still expensive since it is produced in small quantities, but when it will be produced in large quantities, the unit cost will be reduced too much. Internationally, the FDA has approved numerous investigations, new drugs, and applications for different therapeutic applications is there only in research. By the way, I found an very important review article about this topic, published a few weeks ago, and I put it here, and thank you.
Thank you very much. I'd like also to add that our professor is also the vice president of the project dealing with the nano-gold and laser treatment of cancer. These are, a lot of research work here in the National Research Center. Okay, thank you very much.
What has been presented just now is the basics of a big project that has been going on in collaboration between the National Research Center headed by Professor Ashraf Shaaran and the National Cancer Institute headed previously by Professor Ala Haddad and now by Professor Mohammed Boutayeb. This group of research has been under the follow-up or supervision of Professor Mustafa Sayed from the United States, and what has been presented was the basics and the initial lab work on small animals using nanogold and small animals having cancer. Now Professor Ahmed Abdoun, who is a member of the team, is going to speak on the studies and results of applying the nanogold as a therapy for big animals. The group, we worked on about 16 big animals, and now he is going to present the data of this, and after this the floor will be open for discussion. تفضل دكتور.
Assalamu alaikum. In the last time presentation, I will talk about the application of nanoparticles in medicine. So it can be used for drug delivery, gene or immunotherapy, and imaging. And then we will talk about the photothermal therapy using the gold nanoparts and the results of our experiments. So nanoparticle can be used for the imaging, so it's used in the big CT scan and in MRI. Also, it can be used as a drug delivery by conjugation of chemotherapy to the nanoparticle. Also, the nanoparticle can be used as an adjuvant and vaccine delivery or in gene therapy. Nanoparticle can be administered by two ways, by the intravenous injection, and it reaches to the tumor cell through the extravasation of nano system, through the gap, the infiltration through the peak of the gap junction between endothelial cells of the lining epithelium. or it can be administered by intratumoral injection, so it localizes in the tumor cell itself. After injection or administration in the tumoral, the nanoparticle goes to the tumor cell by endocytosis, and after exposure to the laser beam or laser, this nanoparticle become heated and it deliver or produce heat that kills the cell. So after it enters the cells, it increases the reactive oxygen species and nitric oxide, and then it leads to the damage of DNA of the cell, leading to the death of cell. The work that was conducted in the National Research Center on the application of nanogold in pet animals. We worked on the mammary tumor and the skin tumor on 16 animals with mammary tumor and 5 animals with skin carcinoma. These some animals showing the tumor, mammary tumor in dogs. Also, here the mammary tumor, this animal with 2 teeth with tumors. These are some control animals that used for the survival calculation of the survival rate. So this animal kept as a control animal. Some animals also with skin carcinoma. Some cats with mammary gland tumor. So before we start the experiments, we make the clinical examination of the animal. So we measure the blood pressure, the heart rate, and also respiration rate, the temperature and body weight to calculate the dose of the nanoglobes that would be injected to the animals. Also, we take the measure of the tumor, the length, width, and the depth of the tumor, also which was confirmed later by the ultrasound examination. After that, we collect blood samples for the complete blood picture and the liver and function test. This was conducted before administration of the nanoglobe particle and after 15 and 1 month after treatment. Also, we collect biopsy samples for the pathological examination, and after confirmation of that the animal was suffering from grade 1 or grade 2 of the cancer, we start the treatment of the animal. We start first by the injection of the calculating the dose of the nanogold drugs, and then we dilute it in PBS solution according to the body weight of the animal. Then we will start to inject at the base of the tumor, then infiltrate the dose in the different parts of the tumor itself. After 10 minutes for the infiltration of the nanogold drugs to the tumor cells, we expose the animal to the laser for 10 minutes. This is divided according to the size of the tumor itself, so we make different sites for exposure. For the response, from the 16 animals treated with the nanogold drugs, 16.2% of the animals was complete remission within 15 up to 1 month, and there is a partial remission in 25% of the animal. No response in 2 doses, around 12.5%. The tumor relapse in 25% of the animals for animals from 16, and the overall survival rate is 15 days compared to 20 days in the control animals. For the skin carcinoma, there is 60% response of the animal to the treatment. For toxicity study, we collect 2 blood samples. The first one is on ELISA for the CBC, and the other one is a plan for the liver and kidney function. So for the liver function, we measure the total bilirubin, dietary bilirubin, GOT, alkaline phosphatase, albumin, and for kidney function, we measure the blood urea, serum creatinine, and uric acid. There is no significant difference between in these parameters between or after treatment. For blood profile, also with complete blood picture, there is no significant difference in treated animal before or after treatment, after 1 month of treatment. This is in dogs and this in cats. This case, it was the first case that we treated, and here after injection of the tumor, after 5 days, the complete sloughing of the tumor mass after 5 days, and with complete remission of the tumor mass after 21 days. This also, after treatment of intratumoral injection, there is a complete relapse of the animal after 1 month. Also, this animal with tumor in mammary tumor, 2 mammary tumor, one we inject with nanorods, and there is complete remission within 1 month. This case, it was a 4th stage of mammary cancer, mammary tumor, and after we injected, there is 30% remission after 3 days. And after 21 days, the complete remission of the tumor. This cat was suffering from a huge mass of tumor around 14 centimeter by 10 centimeter, and this was 10 centimeter. And after 2 sessions of nanogold injection, there is a complete remission after 1 month of treatment. The animal restored all the biological functions of the activity and fertility and so on. So it became pregnant after 2 months from the end of treatment, and interestingly, the same nerve of the animals producing milk, so they can feed the animals from the same teat. This work was published as a case report in the Journal of Nanomedicine and Nanotechnology in USA with impact factor 4.7. For skin carcinoma, this animal was scrotal and testicular carcinoma, and after treatment with intratumoral nanogold injection, there is a complete relapse of the tumor after 1 month, and the animal still survived up till now, since more than 1.5 years. This animal was tail carcinoma, and this after injection from one dose of nanogold injection, this is a complete remission of the tumor after 1 month. Thanks to the research team and for Pastor Care Foundation for the financial and NRC for the financial support. And thanks to Professor Mustafa Said who guided us during this work, and to my research team. And thank you.
Thank you, Professor Ahmed Abdul, for this presentation. And now I think the next phase of the project was to inject the nanogold to study the acute and the chronic toxicity in normal big animals that are not having cancer, so as to be sure that there is no acute or chronic long-term toxicity in this big animal. If this happened, and after the presentation of this results, there will be soon a manuscript that is going to be sent for publication, and I think after discussion at the National Cancer Institute, and after finishing all of these data, we would have the chance to start phase 1 clinical trial, but not yet until we have the first publication of the efficacy and the acute and the chronic toxicity in the big animals. Now the floor is open for comments or discussion. I hope that this will not take more than 5 minutes in order to keep in time.
Hello. I have two questions. The first one is basic one to Dr. Abdul. Why gold? Why nano gold? Why not nano copper, nano iron? Why gold itself? The second question, you are using phototherapy to sensitize the nano gold. So what you will do in the deep tumors, like GI tumor and other tumors? Thank you.
For the first question, the nano gold is specific for the tumor cell, so it can directly recognize the tumor cell and goes ahead using to it.
So you did it on other materials like iron, copper. Is there any trials on other?
No. We concentrated on non-gold. Yeah.
So what will do for the deep tumors?
Or there is a recent goal which is conducted in China. They use the photothermal therapy for more thermalization, and it can use it for the carcinoma, ovarian carcinoma, and internal organs. So it can use by intravenous injection. Then the cases can be exposed more than one time for the laser, and it could reduce the recovery.
May I add that we have first to have the proof of concept that this kind of technique is working, and my personal suggestion is when we decide to start the human studies, to start on cases, for example, having locally advanced breast cancer that are not metastatic and that are received many lines of hormonal or chemotherapy or targeted therapy, and that you have nothing to do for them. So this could be the start of, if this is proved and there is no toxicities as suggested or expected by the results in the big animal, then we could move forward to idealize the technique. For example, you can use endoscopic approach to inject the nanogold and so on. So it's very, very early to comment on this, but at least we have to prove the concept first. Then we can discuss this later.
Excuse me, I can add here that dealing with photothermal therapy, we can let the nanogold, and this work started I think 15 or more than 10 years ago with Professor Mustafa Said. They started injecting nanoparticles, producing O2 in the cell. So the nanoparticle itself will lead to kill the cell. So we are thinking about this, but the delivery of the drug into the tumor is still a problem for us. So we have this in our mind.
Okay. Professor Madhat Khafaga will be okay, and then Professor Barber.
There are two questions. First question, did you study a section of these tumors after treatment to find any escape cells that didn't take the nanoparticles? Because this would be a nidus recovery. The second comment would be if you are going to apply this in humans, the phase 1 study, I think the perfect example is the xerodermia pigmentosa, because it's a very small tumor and very recurrent, and it's proficient. And so although the cases are not that many, but at least you have 5 or 10 cases with anesthesia every year. So this would be very successful.
This is an excellent suggestion, but for your question, I think that the results show that there is a relapse rate of about 25%. Of course, this is not the magic solution to cure cancer, but it's going to be one of the weapons used to this, but we expect that we may have relapses and so on. So maybe Professor Ahmed Abdoun would comment on the pathology samples that was taken after applying the nanogold.
Actually, we did not take the biopsy samples because there is complete remission, so there is no any superficial signs for the tumor. But we checked the animal with ultrasound to check if there is some roots for the tumor or not. We tried to use the CT scan with the animals, but unfortunately we failed to do such work.
Professor Van Merwe would be the last one.
Congratulations with this very nice preclinical work.
Thank you,
Which is a bright future for the treatment. I have a methodological question. Your controls, were they also irradiated with the laser?
No.
Why not?
Because this is work was all conducted before, so laser receptor did not give the same rate of recovery as with the gold network.
It would be advisable if you want to publish this.
Yeah, we can take it into consideration.
Dr. Ahmed.
Thank you very much for these encouraging preliminary data that will help definitely in the design of future human studies. You have showed the results from cats and dogs, and it seems that within each species there is some differences. I mean, a tumor in the dogs is more sensitive than the cats, at least that one I've got from the slides. Is there any explanation why some tumors responded in dogs, not in cats, something like this?
Yes, it depends on the type of tumor. So with the adenocarcinoma, the response is very high. If the tumor goes to fibrosarcoma or something like this, the response is very low. Some cases which is not responding to totally, the cells is completely changed from glandular cells to fibrous cells.
So it's a matter of the type of the tumor, not the species.
Yes.
Thank you very much, both of you. And actually, it is, unfortunately, we have to finish or to end this session by the schedule which is going to continue after us. Thank you very much.
Now we invite the following chairpersons to